the main focus of my laboratory in the area of cell therapy is to try to control cells following transplantation. And so we've been developing techniques to control the distribution of cells following delivery, as well as trying to control their phenotype and fate following transplantation. And we've also been more recently trying to develop a better understanding of what's actually happening to those cells, and are they differentiating, are they remaining the same following transplantation. And that's what brought us to this most recent paper, which focuses on chemical conjugation of sensors to the cell surface. And these sensors are very versatile, and so we can potentially develop sensors for any ligand of interest, and be able to detect with very high spatial and temporal resolution cell signaling events that are occurring in real time at the single cell level, both in vitro and in vivo. One of the potential applications that I'm really excited about would be to apply these sensors to the surface of cells where the cells have natural homing capability to particular niches in the body. So for example, hematopoietic stem cells, when they're systemically delivered, they will home to the bone marrow. And this is really the basis of bone marrow transplantation or peripheral blood stem cell transfer or cord blood transplants. And so systemically administered hematopoietic stem cells home to the bone marrow, and they occupy a niche which is called the hematopoietic stem cell niche. And so what we're really excited about is taking cells like hematopoietic stem cells, chemically immobilizing sensors onto their surface, having the cells bring those sensors into these niches, and being able to have or elucidate the biology of cell signaling within these niches with potentially unprecedented spatial and temporal resolution. How else would you be able to get a sensor into a particular niche? And so we think by delivering sensors on cells that can home to particular sites in the body that are amenable to intravital microscopy in vivo, that this really offers the potential to better understand what's actually happening in the stem cell niche with great spatial temporal resolution.